Dear 12th class students, today in botany we are going to discuss Megasporogenesis and the structure of female gametophyte as we are continuing with the sexual reproduction in flowering plants. The Megasporogenesis. This is simply the process of formation of megaspores from the megaspore mother cell, abbreviated as MMC. A single cell is differentiated from the ovule, which acts as the megaspore mother cell in the micropylar region of the nucleus, the nutritive tissue located within the ovule. The megaspore mother cell is large with dense cytoplasm and a prominent nucleus. This megaspore mother cell undergoes meiotic division that results in the production of 4 megaspores. As you may recall, that meiotic division results in the formation of 4 cells from a single cell with the chromosome number reduced to half. Now, the female gametophyte. Out of these 4 megaspore mother cells, only one of the megaspores is functional, where the other 3 degenerate in majority of the angiosperms. That means only one of the cells remains functional. And this functional megaspore develops into the female gametophyte, commonly known as the embryo sac. Here you can visualize in the diagrams. This is the cell that gets differentiated as megaspore mother cell, and then it divides into 2, and then after that followed by another division resulting in the formation of 4 megaspore cells. And out of these 4 cells, 3 degenerate and only one remains functional. The development of embryo sac from single megaspore is termed as monosporic development. Now, the development of embryo sac. The nucleus of the functional megaspore divides mitotically to form 2 nuclei, as you can visualize here, and followed by subsequent mitotic divisions. And you must remember one thing, that nucleus divide only mitotically during the formation of embryo sac from megaspore mother cell. First, the nucleus divided into 2 diad stage, and then into 4, and then followed by another division resulting in the formation of 8 nuclei, and finally it is laid into the mature embryo sac like this, which I am going to explain now. The nucleus of the functional megaspore divides mitotically to form 2 nuclei. The nuclei move to opposite poles, making the embryo sac 2 nucleate. The more sequential divisions of the nuclei mitotically from 4 nucleate and later 8 nucleate stage of embryo sac. At 8 nucleate stage, cell walls are laid, leading to organization of the typical female gametophyte, commonly known as the embryo sac. 6 of the 8 nuclei are surrounded by cell walls and organized into the cells. The remaining 2 nuclei, called the polar nuclei, are situated below the egg apparatus in the large central cell. The distribution of the cells in the embryo sac is like this. 3 cells at the micropylar end constitute the egg apparatus. The egg apparatus consists of 2 synergids and 1 egg cell. The filiform apparatus lie with the tip of synergids, which has the function to guide the pollen tube to reach the egg cell. 3 cells at the chilazal are called antipodals. The central cell has 2 polar nuclei, and the mature embryo sac is 8 nucleate but 7 celled. And this is the diagram of the mature embryo sac. These are the 3 antipodal cells at the chilasal end, 2 polar nuclei which lie in the center, or this is commonly called as the central cell, and here this is the egg apparatus on the micropylar end with 2 synergids and 1 egg cell. Thanks.